Welcome. I'm Mahir Tazwar. In this presentation, I'm going to talk about the spatial association of the transverse relaxation rate R2 with latency. Latency is a neurodegenerative disorder of the aging brain. It has been associated with lower memory, cognitive decline, and increased likelihood of dementia. The transverse relaxation rate R2 is one of the main sources of contrast in MRI. R2 is sensitive to increased pre-water content and iron concentration in the brain, and thus it can provide information about the neural integrity of tissue. In this work, we hypothesized that neuronal changes caused by latency can be detected by changes in R2. This study included 797 participants from 3 longitudinal cohort studies of aging. Before death, the subjects participated in longitudinal in-vivo MRI and cognitive evaluation. After death, they came to autopsy and underwent ex-vivo MRI followed by histopathology as shown here. In this study, we used data from the second ex-vivo MRI. The participants have a mean age of 90 years with 16 years of education and were imaged using 4 different scanners. Among 797 participants, 184 had mild cognitive impairment and 365 had dementia. From pathology report, latency was rated on 4 stages based on its inclusion in amygdala, entorhinal cortex or hippocampus CA1, and neocortex. Other neuropathologies that have been evaluated include likelihood of Alzheimer's disease, Lewy bodies, gross and microscopic infarcts, cerebral amyloid angiopathy, atherosclerosis, and arteriolosclerosis. R2 maps were calculated by fitting a mono-exponential decay function, Voxel-wise, to the spin echo data. Individual R2 maps were then aligned to an ex-vivo brain template to facilitate statistical analysis. Voxel-wise linear regression was used to analyze the association of latency with changes in R2, controlling for other neuropathologies, demographics, post-mortem intervals to fixation and imaging, and scanner covariates. The analysis was done using FSL's Pound tool with 10,000 permutations and threshold-free cluster enhancement. The resulting P-maps were family-wise error-corrected for multiple comparisons, and the statistical significance was set at p less than 0.05. This figure shows the coefficient map obtained from the Voxel-wise analysis overlaid on a template used in the study. The coefficient map shows the coefficients in voxels that are significant and represent a linear relationship of the latency stages. The result shows that latency has an overall negative association with R2. The pattern mainly involved temporal, frontal, occipital lobe regions, and basal ganglia, including the white matter areas. The strongest effects in gray matter were observed in the amygdala, hippocampus, entorhinal and parahippocampal cortex, temporal pole, insula, putamen, fusiform nucleus accumbens, and medial orbitofrontal cortex. No brain region showed positive associations of R2 values with latency. Since the associations were observed not only in gray matter but also in white matter, we wanted to see which white matter connection showed an association of R2 values with latency. To achieve that, we used white matter connectivity information from the IIT Human Brain Atlas. Specifically, the track density maps from IIT Atlas were thresholded at 5% of the maximum streamlines per connection. Then we found the overlap of thresholded track density maps with portions of the coefficient map that showed significance in the Voxel-wise analysis. The mean coefficient along each connection is presented in the connectivity matrix on the right. The connectivity matrix shows that the strongest association of R2 with latency are in temporal frontal, and temporal basal ganglia connections. In comparison, there is far weaker association on connections to the parietal lobe. To summarize, this study used high-quality data from a large cohort of older adults. Voxel-wise analysis revealed that latency has an independent contribution on R2 changes and has an overall negative association with R2. This pattern mainly involved regions in temporal, frontal, occipital lobes, and basal ganglia, particularly in the white matter areas. In gray matter, the strongest effects were observed in amygdala, hippocampus, and neighboring temporal lobe regions. In white matter, the strongest association was observed in temporal frontal, and temporal basal ganglia connections. This link between R2 and latency can be used to potentially develop an MRI-based classifier that can predict this disease entity. This study was funded by the following grants. Thank you.